So in this lab, we're going to do battery analysis. We're going to make our own batteries, and this is part 1, procedure 1, page 6. There's the actual voltmeter, and there's vial 1, copper bar in a 1 molar copper sulfate solution. And there is vial 2, a zinc bar in a 1 molar zinc sulfate solution. That's the salt bridge. You can see it on figure 10.2 on page 7 in the lab book to see how that's exactly constructed. So we are now comparing vial 1 to vial 2, measuring the voltage. Notice that the black lead, the positive, is on the zinc side, or the red is on the copper side, and that was 1.05 volts. 1.05 volts. That's the actual reading. All right, so now we're going to go from there. See where we're reading. Don't have to do this every time, but if you're going to change where the salt bridge is, you kind of want to rinse off any solution. So it looks like we're going from again the copper 1 molar copper bar and 1 molar copper sulfate solution. So this is solution 3. This is iron 2 sulfate in a it's an iron bar and an iron 2 sulfate solution. Put the salt bridge across that. Let's see, we're putting the positive on the copper, negative on the iron. Looking at the reading, 0.62 volts, 0.62 volts. That is vial 1 and vial 3. All right. It's off the salt bridge again. We're doing the copper solution versus the, now this is an iron 2, iron 3 mix, so I have to have, there's an inactive carbon electrode. That's graphite actually. It's a form of carbon that will conduct electricity. But because both solutions, both of the reagents are aqueous, we have to mix them together and then put an inactive electrode. I think we had 2 mils of the iron 2 plus and 1 mil of the iron 3 plus. Put the leads across that. Trying the positive on the copper, 0.40, 0.40 volts is the actual reading on that. It's the electrode. Now we're going to the concentration only cell. This is going to be where you have copper bar and copper sulfate solution. One side you're going to have the concentrated copper, and on the other side you're going to have the dilute side. So there it is. There's the 1 molar copper. That's the concentrated. And then on the other side you're going to have the 0.01 molar copper. So you can see it's barely blue in that one. So we're going to be able to get voltage just by a difference in concentration. So here we go. So he had the red on the more concentrated, the positive on the more concentrated. We're reading 0.04 volts. 0.04 4 volts. All right. Now takedown's pretty straightforward. You just rinse off all your electrodes. The carbon one you rinse off in water, then you let it soak in a water vial somewhere, and then later the technicians will put it in acid to soak properly and get all the metal out of there. But for you guys in this lab, what you're going to want to do is put that in the put that carbon in a storage just full of glass of water so it can soak. You don't want those to dry out. Now what he's trying to set up now is going to see if he can make a couple of the copper zinc cells, and we're going to put them either in series or we're going to put them in parallel. So imagine that each one of those copper zincs represents one cell. So you have to have 2 salt bridges to build this properly. All right. So this looks good. It looks like he's going to build it parallel. See how there's copper stacked up against copper, and then behind on the left-hand side he's got zinc, and on the other one he's got zinc. So we're going to see how you wire these up parallel. All right. So zinc on one cell goes to zinc on the other. That connects that. And on the other side, copper on one cell connects to copper on the other cell. It's like putting 2 batteries together with both the positives connected. And then we're going to take the voltage off of you can use either one of those. Connect to either one of those coppers or either one of those zincs. All right. Red goes to the copper. That's where the cathode is. Right? That's where reduction's taking place. And then the black lead goes to the zinc side. And so remember, we were reading about 1 volt when you had a single. Oh, now we're still reading at about 1 volt. So what does that mean? The thing that you can't test with this when you're in parallel is that the time that battery will run has now doubled. So I'm just telling you that because there's no way of being able to test that. I mean, if we were running an appliance, running a light, instead of just measuring the voltage, we could see that the battery will last twice as long when you wire batteries in parallel. All right, so now he's going to try and put it in series. So this is a little trickier. So follow the plan. He's going to kind of move it so it makes better sense so you can kind of see the lineup. That's nice. That's a nice way to do it. So basically, you're going to leave the zinc or the copper of one side and end up on the zinc of the other. Okay, that's series. And then, perfect. Yep, so you leave copper on one cell, and then you're going to have to, and it's double the voltage. Now, what you didn't see, it was kind of hidden, but that black lead went all the way down to the zinc on the far left side. So the black goes to the zinc on the far left side, cross over the salt bridge, copper goes to the next zinc. Once you're on the zinc, cross over the salt bridge, go to the copper and go back to the voltmeter. So it's one big loop. Okay, good. He's going to get in and zoom in on that. See that black lead goes from the voltmeter all the way. Yeah, there he goes. Thank you, Jonathan. Goes from the voltmeter all the way down to the zinc. And that doubled the voltage by putting it in series. That's called series wiring. It will double your voltage. Well, I hope this helps you understand batteries, and I hope to see you in lab someday soon.